Hello, good afternoon, students. I think you can able to hear me, right? So let me check the polls first. Okay, I'm just launching the poll. Am I audible and if the screen is visible? If you're having any issues, please let me know. Okay, check your internet connection and make sure that you are connected to active internet. Okay, and also make sure that everything in your background is perfectly all right. Okay, if you still won't able to hear me, just let me know in the chat box, either in the WhatsApp group. We will help you out. Okay. Okay, great then. So we are just waiting for one or two others to join. Okay, once others are joined, we can start the class. Okay, who are already joined in the YouTube live or in the Zoom, please let me know which particular, if you are joining from a chain, which particular chain we are from, or if you are a student, just let me know. Okay, so great. Okay, great then. Okay, so let me start the class. So this is our first class. Already it's 3 o'clock. We don't want to waste any time, right? So we'll directly into the class. Okay, so this is our first weekend class. Let me explain a little bit about it. Okay, and in this particular class, we are starting the biotechnology chapter. And this particular chapter we are starting today and we are continuing it tomorrow. We will try to finish it up in 2 days. Okay, let me explain how this particular works. Okay, so who are already joined, welcome to the class. Please let others know and tell them to join as soon as possible so they won't miss anything. Okay, so this is your new timetable. It contains like what are the subject and what are the particular timings. Based on the orders from the Navodaya Vidyalaya Samiti, the weekend classes for the outside of Karnataka people is, sorry, weekdays classes are shifted to the weekend class. So as you can see here, Saturday you will be having classes from 3 o'clock to 6 o'clock, and Sunday you have the class from 10 o'clock to 1 o'clock. And your whatever the major test or the chapter test will be there, that will be done in the Sunday afternoon. Okay, Sunday afternoon you will be having your test. So as you may already guessed, because we are having very less time in our hand, so strategies also varied here. Okay, so initially our strategy is like to go slow and go understand everything kind of a strategy. Okay, now our strategy is a little bit changed now because we have a very few hours left, right? So even though April looks like a distant thing because the board exam will be in the March of last week or in the April, right? So it looks like a distant thing, but it's very nearby us. Okay, and the NEET examination will happen in the month of May, and even the ICAR also. I mean, in the KCET and all the things also. So other CET exams also. Okay, just try to remember that even though you look like it's 6 months is there or 5 months is there, when you start dividing your chapters, it will become very less. If you observe the biology, you have 38, similar situation in the other subjects also. Totally 11th and 12th, you will be having 97 chapters, I think, right? You need more than 3 months to read. Okay, so if you are not started reading, please start reading. Okay, so in this particular classes, in the weekend classes, what we do is like we will select a particular chapter and we will do a quick kind of a session in this. Okay, so what we do is like we will try to simplify the concept whatever like available in their textbook, and we will remove all the extra lines and we will make it very simple and understandable. Why? Because we are believe that if you understand the concept, okay, that means if you know what's actually the process is happening, then solving the questions will be easy for you. Okay, if you have any doubt or if you have any questions, you can put that in the YouTube chat box, or if you're in Zoom, you can put that in the Zoom chat box. After the class, if you have any doubt, you can directly post it in your WhatsApp group. If you are in the school, if you have any doubt, you want to contact us, you can contact your teacher coordinator who is present in the schools, and those will send the doubts to us and we will clarify. Okay, no doubt needs to be left out. Okay, whenever you complete a chapter, all the doubts need to be covered. Understood? So, okay, then we will move on to the topics, right? So, our first chapter, like in this chapter, like we are studying three important aspects. One is the like genetic engineering, majorly we are discussing about genetic engineering in that introduction, tools, and the process. Okay, the first things when we come to this particular section is the biotechnology. Right? What is biotechnology? Like technology, we know, like engineering and bio. It's like using biological engineering. It's kind of right. What we normally say in a general discussion is like using the biological things or the bio or any living beings, okay, using living beings and changing it or altering it for the usefulness or the welfareness of human man, okay, for the welfareness of mankind, okay, then only we can call it as the biotechnology. Okay, why we given us such a definition? Because we don't want this to, the biotechnology word cannot be, we don't want to use this particular words in somewhere else. Okay, that's why we given such a kind of a what we can say definition. We will see how the biotechnology works and all the things. Okay, before you are going to the processing biotechnology and all the things, I just wanted to know like how much do you guys already know about this particular chapter or this particular. Okay, let me ask two questions. Okay, try to answer the question either in the chat box. Okay, measured in the chat box. That's the only way of our communication for now. Okay, and so the first question is the cutting of DNA at specific locations become possible with the discovery of. The question is simple. The DNA can be cut at specific points. Okay, so with the discovery of what particular thing we able to cut the DNA at a specific point. Option A selectable marker, option B ligases, option C restriction enzymes, and option D probes. Okay, so let me give you 30 seconds of time. I think that's enough, right? This is a very simple question. Who are like no answer. If you guys don't know what is the correct answer, just let me know that you don't know the answer, so I can explain. Okay, we'll see. Anyone? Simple question. You can think about it. Some question. Let me tell you. Okay, normally people will say NEET is tough. Okay, no. Remember, guys, NEET is easy. Okay, NEET is easy for the people who practice the questions. Okay. So even though you are reading, how can I say, like 12 hours a day, okay, it will be no use if you are not solving the question and answer. I think in the first term test, you realize that, right? The student who solved the MCQ scored better than the student who read more, right? Because like solving the MCQ needs a little bit of the what's the strategies and all, and some of the strategies cannot be like told, like how to happen and all. Some things I will help you out. Okay, some things will come by practice only, right? Okay then, Kuldeep. Okay, correct answer. Correct answer. Yeah, awesome. Right. The cutting of DNA at a specific location, is it possible for the discovery? Is it due to the selectable markers? No. Selectable markers only help us to know that where we have to add the desired gene. So this is not the correct answer. Is it about a ligase? No. Ligase is just an enzyme. Okay, this is not the correct answer. And is it probe? No. Probe also part of like a single stranded DNA. This is also not the correct answer. Then what's the correct answer? As Kuldeep said, the correct answer is restriction enzyme. Okay, this restriction enzyme help us to locate and cut the DNA at a specific point. We will learn about this in detail in the next pages. Okay, so we'll go for the second question, guys. This is also super easy. Okay, think about it like which particular bacteria you most heard. Okay, the question is two bacteria found to be very useful in genetic engineering experiments are first option Nitrobacter azotobacter, option B Rhizobium diplococcus, option C Nitrosomonas and Klebsiella, option D Escherichia and Agrobacterium. Which one is correct? I think you guys can answer this question because like one bacteria is there, we will use this particular bacteria so many times. Think about like molecular biology. Everywhere the chapter is completed, right? The molecular biology. Think about that chapter. In that particular chapter, one bacteria we use it most time. Okay. Think about which bacteria it is in this four bacteria. Okay, and answer this question. I'm giving you again 30 seconds of time. Yes. I'm waiting for your answers. Okay. Correct. Correct. One more person have to check. Yes. Yes. Okay. So 30 seconds is over, guys. So we will directly move on to the answer for the people who are like asking, sir, why are you looking in this way? So YouTube live chats are running in this particular side. Okay, that's why I'm looking this direction also. Sorry for that. Okay. So what is the most heard bacteria in this total 5-6 bacteria and the molecular basis of inheritance? Which one it is? Correct, right? That will be the Escherichia. When you know this particular answer, even though you never heard about the agrobacterium, you can answer this. The correct answer is Escherichia and agrobacterium. For the people who are amazing, like what are these particular bacteria? Nitrobacter and Azotobacter, Rhizobium, Nitrosomonas, and Klebsiella. All these bacteria, you heard it, but where in the nitrogen cycle, right? When we are discussing about nitrogen cycle in the mineral nutrition chapter of the 11th, we heard about this bacteria, either aminification to the aminification and the nitrification, all those kind of the denitrification, those kind of the process. Okay, but for the biotechnology, we will use two major bacteria. One is the Escherichia coli or E. coli, and another one is the agrobacterium. Full form is Agrobacterium tumefaciens. So we will learn that in the next slides. Okay. Okay, shall I move on to the topic then? Great. So as I mentioned, when we are talking about biotechnology, majorly we'll discuss about some things or some process where we utilize the living things and modify the living things for our benefit. Okay. In a general way, if I put, okay, biotechnology can be divided into two major types. One is the genetic engineering and another one is the bioprocess engineering. Okay. How it is different? Okay, genetic engineering is the one, okay, where you modify, okay, you are modifying the organisms. What we are doing here in the genetic engineering, we are modify the organism using a different tools. Okay, using a different tool. So in a simple word, okay, I'm going through very simple kind of the sentences here, so you can understand the process. Okay. Genetic engineering, genes. What we are doing the engineering. Okay, we are using the tools, we are editing the genes. Okay, so what we are doing, we are modifying the organism. Okay, we are modifying the organism. Simply put, think about something. I wanted to make a Spider-Man, so I take some genes from the spider and I put those genes in a human embryo, and I created a Spider-Man. Okay, this is called a genetic engineering. What I did, I modify the organism, modify a human, right? So that is the genetic engineering. I can give like, don't believe that Spider-Man actually exists. Okay, I'm just taking a like what is that hypothetical situation. Okay, don't say that, sir. Can we create the Spider-Man, Batman, and those things? Okay, but it's a hypothetical situation. Okay, then what is bioprocess engineering? Okay. When you are doing the genetic engineering, we need certain set of parameters. Okay, the parameters we need to set this particular genetic engineering is called bioprocess engineering. Okay, so or in a simple way, I can say that maintenance. Okay, maintenance of sterile condition. Sterile condition. Let me explain what is sterile condition. Okay, maintaining the sterile condition or optimum condition, optimum area for genetic engineering. In a simple word, I will put it as a GE. Okay, is called as the bioprocess engineering. Let me take a very best example here. Okay, so I think every one of you know tissue culture. Okay, what we do in the tissue culture, we will take a small tissue from the plant. Okay, think about the very small tissue like this. Okay, a cell. Then we keep in a media. So after some time, what happens? Because we are providing a certain nutrition, but this is not easy because whatever the nutrient you are adding is right. This can be attracted by fungi or bacteria, all this kind of thing. If you leave a bun outside or a bread outside, that will be attacked by the fungi, right? Within like 3-4 days. Think about nutrient media. The media, that means media means wherever we are growing the plant or anything. Okay, other organisms. The media contain high nutrients, okay, high sugar level, high minerals level. the bacteria and fungi actually grow, right? So we need to create such an environment that apart from our desired organism, nothing else should grow, nothing else. That is called a sterile condition. Sterile condition means nothing should grow. Okay, nothing should grow. I will take another one example is the operation theater. Operation theater should be a sterile condition. Or think about something, someone went for the operation, okay, and the operation went very well, but the condition is not sterile. Okay, why they are not doing the operation in a general hall? Why they are not doing the operation in the middle of the road? Why? Because everywhere bacteria fungi will be there. That will cause the infection, right? The patient will die. Okay, I won't say may. The patient will die. Okay, infection will happen. That's why a sterile condition needs to be maintained. The bioprocessing engineering will help us to do or help us to achieve that particular. Okay, so these are the major type of biotechnology, guys. Genetic engineering, bioprocess engineering. Genetic engineering for modifying organism, bioprocess engineering to create the situation for the genetic engineering. Okay, very simple. Understood? So we'll go to the next one. First, we will study about genetic modification of an organism. That's very basic. Okay, here I can make it in a 3 steps, which is also given in the textbook. Okay, so what I will do it here is 1 second. Okay, yeah. First step is identification of DNA with the desirable gene, introduction of identified DNA into the host, maintenance of introduced DNA in the host, and transfer of the DNA. It's a project. So, bunch of words. So let me explain that in a very simple thing. Okay. We will take a same hypothetical situation. I need to create a Spider-Man, right? I want to create a Spider-Man, right? What is the Spider-Man ability? The particular human should be there, okay? Then the host is human. I will consider human as a host, right? And now I want to give him an ability, the ability of a spider. That means creating a web, right? This creating web is the ability. What we call it as, this is, what is this? This is a desired character, right? I need this particular character. Understood? So I wanted to create a Spider-Man. Now I need 2 things. Sorry, my video is turned off. I think I'm not sure. Yeah. So I need 2 things here, right? One is I need a host. I'm considering human. Okay. And I need a desired character, and I'm keeping that particular desired character as a creating web. Okay. Then what else I have to do? I have a spider here. Okay, I have a radioactive spider. Okay, we will think about a spider. Okay, so we will imagine this is the spider. Okay, just imagine, guys. Okay, so I'm making it 8 legs, right? Because it should have 8 legs because it belongs to a class Arachnida. Okay, in the class Insecta, right? Sorry, in the class Arthropoda. That's why it should have 8 legs. Okay. Now what I have to do? I have to check its DNA. Okay. Now I have to check the DNA. It have a very long DNA. Think about it, this much long DNA it have. Then I'm trying to find which particular sequence is actually responsible for creating the web or the gland. So when I'm searching, I got to know that this part of the DNA here to here is responsible for creating organ for the web. Okay, creating organ for the web. This particular genes, this here to here, the gene is creating what? This is responsible for creating the web in that particular, what is that? Our spider. Okay. Now, this particular gene is called as the desirable gene. Then what is the first step I have to do? Remember, the first step I have to do is I have to identify the DNA where it is. I identified already the spider in this particular portion. The genes are there which actually gives a web. Okay. Then that is the first step, identification of DNA with the desirable genes. This is the desirable gene, and I identified the location. This is called identification of desirable gene. So, right? This one is complete now. What is the second step? Introduction of identified DNA into the host. What is the second thing you have to do? You extracted now. Think about like you extracted this particular DNA. then you have to transfer this DNA into the host. That means human, right? You have to introduce into the human. This particular process, that means taking this DNA from the spider, okay, and adding into the host is called as the introduction identifying DNA into the host, or the second step. When you added the DNA into the host, do you really think that it will be accept, oh, I got a new DNA, I will get a Spider-Man? The cell never think like that. Okay, cell is like so strong. It always feel like cell, whatever happens, it's a territory. Okay. So what you do, think about like in your home, without your permission, someone came inside. What do you think? You think that he's a thief, right? So what you will do, either you will beat him, right? If some aggressive person is there, that person may kill him, right? So that's the case, right? Our body is also like that. If you take any cell, if you add any external DNA or external RNA, our body won't accept it. It thinks that some thief is coming or some virus is coming. What it will do, it will destroy it, start to destroy it. So what we have to do, before adding this particular DNA or after adding this particular gene or the DNA, we have to create such a particular condition that the DNA should be maintained in the host. Okay, it should be maintained in a human, and that particular what is that genes should be transferred or inherited to its progeny. Okay, that is when you will become successful. Okay, so it should be not like when you add the spider DNA to a human and you will leave it will happen automatically. No, it won't happen automatically. You have to create such a situation that the cell should accept whatever the desired DNA we are adding. Okay, so these are the main steps. You have to identify, you have to process it, you have to add it into the host, then you have to make it maintain. Okay, now 2 or 3 words you have to remember, guys. The host, the first word you have to remember is host. Host means where you are adding your DNA. Okay, second word you have to remember is desired gene. Okay, desired gene or desired character, that means character which we want to see in the new organism what we create. Okay, so these are the 2 major words you just have to remember. Okay, shall I move on to the next one? If you have any doubt, just put that in the chat box. Okay, I will solve and okay. Okay, then we'll move on to the next step. Just I added a picture here just to represent here. Okay, think about like I want to create an insulin because everyone wants the insulin for the which particular disease? Correct. For the diabetes mellitus, right? Diabetes mellitus, in the simple word, I can call it as like sugar disease, right? A simple, we can also call it as a diabetes, right? For us, like everything is same, but for you guys or for us, at least for the science people, you have to know the sugar related disease is diabetes mellitus. Okay, because if you read the human physiology, especially the expression, there is one more diabetes is there, that is diabetes insipidus, where water absorption won't happen properly, right? So this one we are discussing is the diabetes mellitus. So one person is there, he can't secrete the insulin. So I want to create insulin genetic engineering. How I will do it? First, I will take a human pancreas cell because that's where actually our insulin is produced, right? Then I will see here and I will find a DNA. This particular DNA which actually code for the insulin. Can I call it as a desired gene? Yes, right? I can call it as a desired gene, right? This is the desired gene, right? Then what you will do, I will separate it. Okay, I will cut and I will separate it. Okay. Then at the same time, I want to multiply it. Okay, I want to multiply it and I want to add it to the host. For adding into the host, okay, here the host is bacteria, E. coli. Okay, I need a vehicle to that. It's not like I will just throw it in there, right? I need something to transfer. That for that we will use a specific kind of thing called a plasmid DNA. Okay, so I will explain everything bunch in the later. Okay, just as an overview. Okay, don't take tension. I will take plasmid DNA and I will cut this particular DNA in such a way that this particular desired gene come and attach here. Okay, that's how I will cut it. So when I join these two, what happens? It will create a recombinant DNA. What is recombinant DNA? Recombinant DNA means any DNA which is, any DNA where we added our desired DNA or where we modified, okay, The modified DNAs are called recombinant DNA because recombination happened here. This is a different gene, this is a different gene, right? Both of them are combined here. So I will take this recombinant DNA or recombinant plasmid, I can call both. Okay, then I will add this into the E. coli cell and I will grow this E. coli in a fermentation tank. When I grow in the fermentation tank, what happens? Whenever the bacteria is doubling, that means every 20 minutes, what happens? My desired DNA also doubles. Whenever my desired DNA doubles, what happens? It will start to produce human insulin. What I have to do next? I have to separate the human insulin and I have to extract it and purify it. Then I can store it in a bottle and I can sell it to all the people. Right. So this much it is. Biotechnology, the complete thing, whatever we study, right, in this particular chapter, principle and process. Okay. We can put the complete chapter in this much small diagram. Okay. So this much it is. If you are like want to keep this particular diagram, take a screenshot. Anyway, I'm sending the notes also in the WhatsApp group. You can get it from your coordinators. Okay. Let me continue. So the first step, as I see, what is the first step here? The first step is like identify. I identified already DNA. I have to separate it. The separation of DNA is called isolation of DNA. What I'm doing here, okay, I am isolating the DNA from the cell. Okay, I'm isolating the DNA from the cell. Right, I'm isolating it. Right. So this is called isolation of the DNA. So is it easy? Or is it tough? How it should be, right? Remember, I think both of you know that, right? So there are different processes will happen, right? So what will happen here? There is a cell, and the cell will be having. If it is a plant cell, okay. If it is a plant cell, what happens? It have a cell wall. What cell wall? A cellulose cell wall, right? If it is a fungi, then it also have a cell wall. What cell wall it have? It have a chitin cell wall, right? It have a chitin cell wall. Think about it's an animal cell. Okay, if it is an animal cell or bacterial cell, think about it's a bacterial cell. Then what kind of cell wall it have? The bacterial cell have a cell wall of murin, right? Murin cell wall. So every different types of the cells have different types of the cell wall. If I take animal cell, there is no cell wall, right? Guys, give me 1 minute, okay? So there is some disturbances happening, so I will just join, like I'll be like explain it in a minute. Okay, so let me explain the further. Okay, so if it is from the fungi, what happens? It's have a sorry, sorry. Okay, if it is from a bacteria, then what happens? It will be having murin And if it is a plant cell, then it will be having cellulose, and if it is animal, there is no cell wall, and if there is fungi, it's a chitin cell, right? So I have to remove that part, right? If I want a DNA is present inside here, right? I want to remove the cell wall. So what is the first step? We will use the cell, or cell will be lysed. Remember, this is also another term you have to remember. Lyse or lyse means break. Okay, when you break. Okay, so you can say lyse. Okay, lysis. Actually, the word is lysis, but here you use as like as a term. Okay, lysis means what? Lysis means break. Okay, I have to break it. How I can break it? I can break it using enzymes. Okay, there are different enzymes we normally use. For the chitin, we will use the chitinase, and cellulose, it's cellulase, and for the murine, it's a lysozyme. An animal, anyway, we know we don't need anything like that, right? Understood? The remembering these particular enzymes also super easy, because chitin is chitinase, you just have to add ASC, cellulose, cellulase, right? The murine is the only thing which is different. It's not murinase, don't say like that, it's a lysozyme. Okay, lysozyme. So I break it now. Okay, I broke it, just I have a plasma membrane. Next, what is the next step to get the DNA? I have to break the plasma. How I can break the plasma membrane? I think some of you already did the experiment of DNA extraction, right? What we will do, we will add a specific kind of the chemical, especially in your lab. While you are doing it, what you guys did, you guys added the washing liquid, and also you guys added the NaCl, right? So in the similar fashion, when we are extracting in biotechnological process, we will add some chemical which will normally break the plasma membrane. Okay, because plasma membrane are made up of lipid membranes. If you add certain detergent kind of materials, the lipid membranes will break up. Okay, so what is the next step then? Okay, I have now DNA along with some proteins and different RNA and everything, because you know how the DNA is arranged in a molecular we studied, right? It's combined with everything. What is the next step I will use? I want to separate the protein. What I have to use? Think about it. Just have to ask the ASC, right? We will use the protease. When we add the protease will break the protein molecule. Right? And what left then? DNA plus RNA left. Now I have to remove the RNA also. Which one I will use? Okay, I can use RNase, okay, or ribonuclease, okay, ribonuclease, or I can also call it as the RNase, okay. This will break what? This will break the RNA. This will break the RNA. So we're left with our material that will be DNA. Okay, that will be DNA. But this DNA is not clumped together. This will be like, looks like the water only. Okay, you can't make a separation. Then what we will do, we will add a chilled ethanol. When you add a chilled ethanol, what is the meaning of chilled ethanol? Or what is the meaning of chilled? Chilled means what? Cold, right? Chilled means cold. When you add this chilled ethanol, what happens? This DNA will clump together and it will form structure like this. Okay, I think whoever did the experiments are already know like this, right? White color, the substance will come. We call it as a DNA. Sorry, it's a DNA, right? So your DNA is now ready. Look guys, very simple. As I said, first step will be breaking the cell wall. You can use cellulase for plant cell and chitinase for fungi cell. There is no need of any enzyme for animal because there is no cell wall for breaking. Okay, and the lysozyme for the murine, right? Once the cell wall is broke, then what you have to do, you have to break the plasma membrane. We can break the plasma membrane using the different detergent kind of the materials, because these are made up of the lipid layer. Okay, after breaking the plasma membrane, we left with DNA along with proteins and RNA. What we will do, we will add the protease, the protein will break, then we'll add the ribonuclease, which will break the RNA, then we'll have a DNA. But when you add the chilled ethanol, the DNA will DNA will separate and you can be taken out. This much easy it is. Okay, simple. You just know the process. Just Put it in a flow chart. Remember, if you put it in a flow chart, it will become easy like anything. It will become super easy. Okay, it will become super easy. Now think about something. Okay, so we have a DNA here. Okay, I can understand, right? But this DNA, can we utilize properly or directly? No, this DNA is very long. Okay, this particular DNA is very long. Okay, several meters long. You can't do anything with that much of DNA, right? Because think about a small thin rope, which is like 100-meter long. You want to like drag it. What happens? Everything will break. So what we have to do is we have to make this long DNA, this much long DNA into small pieces. There our next step will come. Okay, so that we will see after this particular question. Okay, now the question comes, guys. Try to answer the question, and if you don't understand anything in the isolation of DNA, please let me know. I will help you out. Okay, which of the following is not correctly matched for the organism and its cell wall degrading enzyme? Okay, the question is simple. Which particular enzyme is not linked properly? Option A, methylase. Option fungi, chitinase. Option C, option C, bacteria, lysozyme. Option D, plant cell, cellulose. This question is asked in NEET 2013. Okay, you can answer or start answering in the chat box. I'm repeating the question. Which of the following is not correctly matched? Okay, whenever you are see the correct, you just start to check 2 times, because as I've seen, most people or most students will make mistake by thinking that it's correct. Okay, so they are not asking about correct option, they are asking about not correctly matched. Okay, matching is not correct. Okay, we'll see. I'm getting the answer. Some people are saying A, some people are saying B, some people are saying C. We'll see. Okay. 30 seconds is over. I think we can reveal the answer. Right, we will go by down. Okay, from the plant cell we will come. Yes, plant cell is comprises cellulose. The plant cell wall is made up of cellulose. The cellulose breaking enzyme is cellulase. So this option is correct. So this is not the correct answer, right? This matching is correct, right? This match is correct. Then bacteria cell wall is made up of, as we mentioned, murine, right? It's made up of murine. It can be broken down by an enzyme called the lysozyme. So this match is also correct, but that's why option is not correct. Remember, bacteria, the enzyme is lysozyme. Okay. The third one is the fungi. Fungi cell wall is made up of what? It is made up of the chitin. So it is degraded by enzyme chitinase. I told you, right? Cellulose, cellulase, chitin, chitinase, right? So this match is also correct. So B is also not the correct answer. Then what is the correct answer? The correct answer is algae. That means the match is not correct. Let me explain you why the match is not correct. You know that algae belongs to which kingdom? Algae belongs to which kingdom? Correct, plant kingdom. As I mentioned here, plant cell should have which cell wall? It should have the cellulose cell wall, right? So if it is having cellulose cell wall, then what is the enzyme it should need? It need a cellulase enzyme, right? It need a cellulase enzyme. But what they given? It need a cellulase, but they given a methylase. Methylase is the enzyme which add methyl group. That is not the correct answer. That is not the correct match. Okay, that's not the correct match. That's why A option is correct. Okay, A option is correct. If you have any doubt, please let me know. I will explain one more time. Okay, shall we move to the next thing? Next thing, we will move on to the nucleus. Okay, okay. Now what I need, I have a very long DNA I need to cut. Okay, but when I'm cutting, I need to know where I cut also. Okay, it's not like I randomly cut here and there. I just want to know where I cut the DNA. Okay, so there we come to know about a particular kind of enzymes called nuclease. Okay, enzymes called nuclease. These enzymes, okay, these particular enzymes does not means they will divide the nucleus or anything. Okay, please remember that nuclease, that does not mean that they destroy the nucleus. Okay, nuclease means they cut the DNA. Okay, nuclease means enzymes. Write down if you want enzymes which, one second, which cut the DNA molecule. Okay, so the discovery of this particular is also very what we can say nice. Okay, what happened is in 1963. Okay, in 1963, the scientist, 1963, scientists working on Escherichia coli that we mentioned, right? What we are doing is like they are working on the E. coli bacteria. Okay, they are doing their work on E. coli bacteria and how the bacteriophage will affect on that. Okay, bacteriophage will affect on that. I think everyone of you know what is bacteriophage, right? What is bacteriophage? Bacteriophages are virus of bacteria, right? Virus of bacteria. What happened is like when they are observing that, they come to know that there are certain enzymes in E. coli that will restrict the growth of the bacteriophage. So normally bacteriophage should affect the E. coli and the E. coli should die. Okay, but there are certain enzymes in E. coli that's actually stopping the growth of the bacteriophage. When they seen this, they discovered that there are two types of enzyme present. Okay, there are two types of enzyme present. One enzyme is adding methyl group. Okay, one is adding methyl group to bacteriophage, okay, group. This particular what it's taking is taking the DNA, okay, and it's adding methyl group. I think this particular group, you know, right? This methyl group to the bacteriophage DNA. Bacteriophage DNA. You may ask, sir, what is the need of adding methyl group? Guys, just check here. What is the valency of carbon? 4, right? When carbon in 4 valency or when it's filled, what it is become? It will become less reactive, right? So remember, in biology, wherever the methylation happens, okay, methylation means DNA or anything. If the methyl group is added, that's called methylation. That means what? The reaction is stopped. Okay, this particular DNA cannot react with the other DNA or add to the other DNA because methyl group is added. So what this particular bacteria will do, think about this, the bacteria, and here our bacteriophage DNA, here it's our bacteria DNA. Okay, let me explain. It's a bacterial genome, and this is our bacteriophage DNA. Okay, what our bacteria is doing, it has certain enzymes. One enzyme go and add methyl group here. Okay, add methyl group to DNA. So because of that, what happens? This bacteriophage DNA cannot bind to the genome. Okay, bacterial genome. Because why? Because it added the methyl group. Understood? Why it can't join now? Because it added the methyl group to the end. Understood? So this is the first type of enzyme they discovered. The second type of enzyme they discovered, instead of adding methyl group, which is actually cutting, it's actually cutting the DNA. Okay, which is actually cutting the DNA. Okay, so this particular enzymes are called as the nuclease enzymes. Okay, these particular enzymes are called as nuclease enzymes. When they started reading more about this enzyme, okay, when they started reading more about this DNA, which actually cuts, so they discovered that some enzymes in this category cut the DNA in the end. Okay, cut in the end of DNA. End of DNA. Okay, they normally cut the DNA in the end. Okay, that means this is the DNA. They normally work in this end. So these enzymes are called exonuclease. I think you understood, right? Exo means what? Outside or far side, right? Exo, that means outside, not in the middle. Outside they are cutting. Those are called exonuclease. Okay, but certain enzymes are cutting in the middle. Okay, cut in the middle of DNA. Cut in the middle of DNA. The enzymes which cut in the middle of DNA, they call it as the endonuclease. Endonuclease, endo means what? Endo means inside, right? Or in the middle, right? What they are saying? Nuclease, that means cutting the DNA, which cut the DNA, right? Where it is cutting? It is cutting in between the DNA, right? In between the DNA, these are called endonuclease. Okay. as the endonuclease. When they started working on that, they will also discover that this endonuclease can be again divided to two types based on how they cut the double-stranded DNA. Okay, there are two types of cutting happens. Certain enzymes cut the DNA exactly in the middle and forming this kind of DNA. Okay, this kind of double-stranded DNA. Okay, for some DNA, sorry, some enzymes cut the DNA something like this. Okay, leaving like this. Okay, where the DNA cut happens in the exact middle. Okay, and having a blunt end called as the blunt end DNA. Okay, blunt end. Blunt end DNA means what? Here it will be looks like this: G, C, and A. T, right? This is a blunt end, right? Here it's continued, right? This is a blunt end. But where DNA having extra, okay, extra one stranded is called as the sticky end. This is called as the sticky end DNA. I will give an example. Sticky end DNA looks like this: AT, GC, okay, like this, and A, sorry, T, G, C, A. Okay, here what happens? This particular nucleotide wants to form a bond, right? If you give something like CGA here, if you give anything like CGA which time, they will come and join, right? They will join like this because they are equal and opposite. I think you learned this in molecular. So that's why we call it as a sticky DNA. Understood? Remember, nucleus means DNA, sorry, enzymes which cut the DNA. We first discovered in the 1963 in E. coli. Using bacteriophage and E. coli model, we find two types of enzymes. One add methyl group to the end, okay, or we can say methylation process. Another actually cut the DNA. This DNA which cut actually have two types. Some cut at the end of DNA called as exonuclease. Some cut at the particular point in the middle called endonuclease. These endonuclease again divided into two types based on how they cut the DNA. If they cut straight, they are called blunt DNA. If they make or if they leave one strand extra, it's called as the sticky end DNA. Okay, so any doubt, guys? If you have any doubt, please let me know. Okay, please let me. Okay, shall I continue? Okay, so we will go for the little bit further about this endonuclease. Okay, there are certain endonuclease enzymes which cut the DNA at specific point. Okay, specific point, exactly at that point. We call them a restriction endonuclease or REN. Okay, what is the meaning of restriction endonuclease? Restriction endonuclease means enzymes. Endonuclease means N should be capital, right? N should be capital. So restriction endonuclease means enzyme, or I can in a simple word put it, any endonuclease which cut DNA, at specific point. Okay, at specific point. It will know all the time, it will cut at the same point. It's called the restriction endonuclease. Remember, restriction endonuclease are very, very important to us. Okay, we discovered that this restriction endonuclease enzyme cut any DNA at a particular site called as a palindromic sequence. It will cut, RAN will cut the DNA at a specific site called palindromic sequence. What is the meaning of palindromic sequence? Do you guys know? What is the meaning of palindromic sequence? Palindromic sequence, correct. Palindromic sequence means any sequence. Okay, give me 1 second. Okay, so this particular palindromic sequence means any sequence which actually is repetitive. Okay, let me take a general example first, then we'll go for a DNA example. I will take an example of Malayalam, or I can take an example of I, right? Here what happens? If you read from this side to this side or this side to this side, the meaning is same, the reading is same, right? I, this side to this side, this side to this side, Again, meaning is same. So we'll take a DNA example. I think you understand what's the palindromic sequence. Let me take a DNA example. In DNA, I will write something like this. Here on it's GAATTC. This is the 5 prime to 3 prime. I think every one of you know what is the meaning of 5 prime to 3 prime, right? Again, 3 prime, it will be CTTAAG. So it's a 5 prime. Check about this particular sequence, guys. When I read, that means not me, polymerase. Okay, polymerase, you know that the polymerase enzyme always read in 5 prime to 3 prime direction, right? Always read in 5 prime to 3 prime direction. When it is reading this strand, okay, it will read as GAATTC. Okay, then that polymerase enzyme again reading the other strand, you know, right? Where to where it will read 5 prime to 3 prime. Again, it will read GAATTC. So what's happening for the polymerase? Whenever it's reading in forward direction or the backward direction, it is reading in the same manner. This kind of sequence where the same sequence is repeated in the both template strand and the coding strand are called as the palindromic sequence. Okay, this particular restriction enzymes only cut in the palindromic sequence. Okay, so let me take an example. There is one restriction enzyme called E-core-R1. Okay, you may be amazed, sir, what is this word means or how this particular name comes from, right? The name because comes from a particular orderly fashion. I'm writing down here E-core-R1. Every word represents one particular thing. Normally, the first word of the enzyme represent the genus. Okay, the genus from where we extracted. In this case, it is Escherichia. Escherichia. I told you right. Initially, only we use this Escherichia coli too much times in the biotechnology. The two words in the later, okay, the following the genus letter, the small letter words normally represent the species. Okay, here in this case, coli. Okay, the third big letter will represent the strain. I think you know about the strain, right? So here it is R or the rough strain. You know about rough strain and a smooth strain, which chapter? Molecular basis of inheritance, right? In Griffith experiment, right? Same like that only strain, it represents the strain that is rough. So what is this I represent? No, it's not I. It is actually a number, okay, which is actually a number or order number we say, okay, order number. Don't think that the scientists are always successful in their first thing, okay. There are like from a single enzyme, think about E-core, Escherichia coli, I will extract 100 enzymes, 100, think of it's a hypothetical, okay. 100 restriction enzyme or endonuclease I will extract. Then how we name it? We will name it by the extraction order. That means what? E-core-R1 means this particular enzyme is extracted from Escherichia coli or strain in the first order, in the first number. Okay, that is the particular name represent. Okay, when you take this particular E-core-R1 as an example, if you put on the DNA, what it will do? First, it will identify where is the sequence which is needed to that. E-core-R1 have a specific site or a palindromic sequence needed, GAATTC. Remember guys, this is the one which is given in your textbook. That is why you have to remember the palindromic sequence of E-core-R1 enzyme. Okay, so what it will do? It will go and find the sequence where GAATTC is there. And what it will do? It will cut this DNA in after the G and again here after the G and it will cut here. Okay. So what happens here? Because of this order, what happened? We got two DNA, right? Sorry, two cut of the DNA, right? The one DNA is divided into two cuts. So this particular cut, as I seen, is it a blunt or a sticky end? Which one you guys think? Is it a blunt or a sticky end? Correct. Because it looked like this, which particular end it is? This is the sticky end. Okay, this is the sticky end. Sticky end. Understood, right? So it's producing the sticky end. So you understood GAATTC for E-core-R1, it is giving the sticky end because it's cutting the away from the middle, right? Away from the middle. Can we see some other examples for the other bacteria, sorry, enzymes? Look here, okay. I have a chart. This chart explains the enzyme name and where they extracted from and which particular site it will cut and what type of ends you will get. First one, E. coli R1. It is extracted from E. coli RY13, okay, and which can be seen cut in GAA-TTC, right, and it will create a sticky end. But the first restriction enzyme, what we extracted, it's not this one. Actually, that is Hind2. Okay. The first restriction enzyme we got is the Hind2. Here I'm not given Hind2 because that's not there. Okay. We will take an example of Hind3. As I said, H means what? H means Haemophilus influenza. Okay. Hind. And D represent what's a strain. Okay, and what is the 3 represent the order of its extraction. It will also create a sticky end, but its sequence are different. Look here, its sequence are EAA, GC, TT, and it also cut after the A and here, and it will create a sticky end. Same with the BAM-H1. In BAM-H1 what? Bacillus amyloliquefaciens, and H represents the strain, 1 is the order. It also produces sticky and here the codon is GGATCC. Okay, so same with the SAL1, which is extracted from Streptomyces alvus G. It creates a different end called GTCGAC. These are all sticky ends. But let me give you a second example. We'll take BAL1. Okay, we'll take the BAL1. So here, as you can see here, Brevibacterium albidium. Okay, sir, where is the strain here? There is only one strain. When there is no strains are available of the bacteria, okay, when there is no strain is available of the bacteria, we will remove the word. As you can see here, SAL1, as you can see here, BAL1, that means there is no strains, okay, so other than the standard strain, okay. Here you can see the strain or the DNA is TGGCCA. What's happening? It is cutting in the middle. It making a cut here. Because of what kind of end we are getting? We are getting an end like this. What kind of end it is? Blunt end. Okay, blunt end. Understood? So there are two types. Either it will be sticky end or it will be blunt end. As you can see here, which one is more helpful? The sticky ends are helpful because sticky ends are easy to fuse. Sticky ends are easy to fuse. So that's why easy to fuse. That's why sticky ends are helpful as even I'm not saying blunt ends are not at all helpful, but sticky ends are helpful us. Okay. So these are the major things, guys. The time is almost over. Okay. There is one more question is there. We will convert it into homework. Just take a screenshot or just read it. The specific palindromic sequence which is recognized by ECO R1. Okay. So other than that, we will study about other things in the tomorrow class. Before coming to tomorrow class, do this homework. So try to read the biotechnology chapter. Even though if you don't get the time, at least read what are the head points and what are the diagrams or the pictures are given. Okay. And if it is possible, solve MCQ given in the module. The modules will be sent in the WhatsApp group. Contact your teacher coordinator if you won't get the module. Okay. So this is for today. I think you understood the class. Okay. Thank you for joining. Please also inform others to join. If you have any doubt, please put here or you can also text me in the WhatsApp group. Okay. Thank you. We'll meet tomorrow morning. Okay. Bye-bye. Thank you. Thank you, Jane Sangli.